Hi everyone. I'll start the class in just 2 minutes. We'll wait for maybe a couple more people to join. Okay, I think we can start the class today. People will join as we go. So, hi everyone. To those who have not joined before, I'm Srishti. I'm a PhD student at ISC and I'm one of the TAs for this course. And we're going to be just discussing some concepts from your most recent week's worth of coursework from the course, as well as some question answers that will help you understand things better, hopefully, and also tackle your assignments and the exam if you have registered for it. So I'll go ahead and share my screen. And if anyone has any questions during my explanations or during the discussion of question answers, like please go ahead and ask them. Uh, okay, great. So I'll just have a quick recap of the concepts as we go. I'll just share my screen. Oh, right. I hope everyone can see my screen. Just let me know if you can't.
Yes, ma'am.
Yeah? Okay, great. I can get rid of this. Okay, all right. Uh, right. So, hi everyone. Thanks for joining, and we'll get started on discussing the coursework as well as a problem-solving session. And to those who don't know how we typically like to do things here, when we go through the questions, they're mostly multiple choice. So what I do is I show the question, and then you can send the answer in the chat, or if you feel more comfortable, you can also unmute and say it out loud. It's up to you. And then we'll discuss as we go. Okay? So let's get started. So a quick recap, which will hopefully help you with this week's coursework, which is from the last week's work. Okay, right. Yeah, so a quick recap here is as you learned about muscles and the cardiac muscles versus skeletal muscles, you learned that when there is some external electrical stimulus to the muscle for it to contract. Or in the case of the heart, there are two types of electrical activities of the heart. So in these cases, you have these neuromuscular junctions, which essentially are places in the body in between the muscles and the nervous system. They are connections that allow for the passage of electrical and chemical information, right? So as you know what a neural synapse is, right? It's between two synapses. You have electrical activity, and you also have a bunch of these neurotransmitters and neurohormones that are released that also pass through the information. So at the neuromuscular junction, you have a synapse between the neuron and the muscle cell or the muscle tissue, and you have your presynaptic terminal containing your vesicles full of neurohormones or neuroenzymes like acetylcholine, and then you have the ACH receptors on the other side on the postsynaptic membrane that allow for ACH to bind and pass through information, right? So this is the neuromuscular junction. This is it in slightly more detail, right? And then once you have this information, what happens, right? So you have membrane depolarization, you have calcium influx from both in the case of the heart, from the cytoplasm, and then a little bit from the sarcoplasmic reticulum, right? And then that goes and talks to actomyosin. So it talks to actin and myosin. You have your Z discs over here, and the calcium helps to displace tropomyosin to allow actin and myosin head binding, which allows for contraction. So this is happening in skeletal muscles. It's also happening in cardiac muscles, right? So all of this is orchestrated by information that is crossing through the neuromuscular junction, right? So this is what we learned. And then, as you can see here, there are these nicotinic acetylcholine receptors, right? So as we talked last time, when it comes to a connection between, from the medulla oblongata all the way to the heart, you actually have two types of receptors. You see nicotinic receptors and you see muscarinic receptors. So muscarinic receptors are there on the heart muscle in the SA node and so on, and the nicotinic receptors are the preganglionic ones. So nicotinic receptors are also an acetylcholine receptor. They are one type of acetylcholine receptor, right? And they respond to this neurotransmitter. And nicotine, it's called that because nicotine can bind and it act as an agonist for the receptor, right? And it's present at the neuromuscular junction, and it is very important for motor nerve to muscle communication, right? And so we see it in the heart, um, in the pathway to the heart from the medulla oblongata. And the type of receptor that it is that it's ionotropic, meaning that it is directly linked to ionic channels that allow the passage of ions upon acetylcholine binding and some conformational changes, right? Then the next type of receptor we learned about are the muscarinic receptors. So muscarine is the agonist over here that binds to this receptor more specifically. And this is also an acetylcholine receptor. It's a different one, right? And it is found in the heart and on certain other cell membranes. And it has a whole bunch of roles in the postganglionic fibers. And this one is not an ionotropic receptor, it's a metabotropic receptor. So it's a G protein coupled receptor, meaning that it works through the GTP-GDP cycling of G proteins. And that's what allows it to have conformational changes that allow for then information cascade to begin within the cell after acetylcholine has bound. So in this way, we learned about these two types of receptors that help with electrical information to reach from the brain to the heart. The heart also has its own individual electrical activity started at the SA node, which is your sinoatrial node, and that conducts electrical activity to the AV node or the atrioventricular node, and then that conducts the electrical impulse further into the ventricles via the bundle of His, um, and this allows the heart to then contract and uh, maintain its rhythmicity. Okay, so we learned about the heart and these types of electrical activity, and then we covered hemodynamics. So the just the starting part of it, and then this week's session will cover more in detail about hemodynamics. So we learned about how blood is distributed in the body in a very clever way, where it's if we have 5 liters of blood in our body, it's not actually possible for all the blood to be everywhere at all the time. So the body really needs to prioritize. So there are clever ways in which the body has figured out how to redirect blood to where it is most required at that time. So there are different percentages of the blood at homeostasis or at like baseline levels. Imagine you're like not running away or you're not exercising or doing something, you're just sitting, relaxing. That time you will have these kind of readings, right? So the blood is leaving your heart, the left heart, at quite a high pressure, relatively speaking, because you have it being pushed by the into the aorta by the strong contraction of the left ventricle, which has a very thick musculature, and this blood then moves into the lower regions of your body, all the way from the thorax to abdomen to legs, everything, right? And so you have a whole bunch of blood, you have 5 liters of blood per minute uh, circulating at this high 100 mmHg. And then this distributes into different organs. And how do we decide, right? So there is electrical and chemical activities that decide which organ needs it, and you have your parasympathetic and sympathetic nervous systems also coordinating which organ needs it. For instance, if you are running, exercising, more blood is redirected to your skeletal muscles and less to your GI system. Whereas at equilibrium, you typically have somewhat a balance where you have blood going to your GI system and less to your skeletal muscles. And it goes the other way around when you're sleeping. So, like that, you have this clever redistribution of blood based on requirement of the body at that time. And obviously, to maintain this and maintain these pressures, there's a lot of um, dynamics at the level of um, not just at the arteries and veins, but also at the level of the capillaries, where individual capillaries have sphincters that allow or don't allow blood into a particular organ. And so some capillaries are active and some aren't. You have veins which prevent backflow because they have valves, and then you have arteries that manage this pressure of systolic and diastolic pressure by having this thick musculature that allows for all this contraction. So all these things put together are maintaining the blood pumping through the body at the right pressure in the right region, right? So then after that, we also did a little bit of a math problem last time, which was a simple one, which was how to calculate the mean arterial pressure. So we saw that if one example, right, where this person who has unfortunately slightly low BP, right, this person has 110 by 60 mmHg BP reading. So what is their mean arterial pressure? So if you remember, um, this is just for your recap, but like the mean arterial pressure formula is 2 into the diastolic pressure. So what is the diastolic pressure? Is this, right? The lower one, plus 1 into the systolic pressure, which is this one. Basically, when your ventricles are in systole, that's your systolic pressure of your blood, divided by 3. So that's what it is. So if you were to calculate this, how much would it be, right? So you have 2 into 60 plus 110 divided by 3. So this person's mean arterial pressure is 76.6. So like that, we had done one more problem also last time. You can always refer to my uploaded slides. Right. So coming to what is new this time, we'll talk a little bit about hemodynamics. So I think this is pretty well explained so far. I thought before we go into question answers and these concepts, I thought let's cover something slightly interesting and slightly different and exciting that I thought would be nice to look at before we jump into what you already know. So that is the question of adaptation. right? Environmental adaptation. How do people under extreme climates and conditions cope with low oxygen tension? So where is the low oxygen tension, right? On top of a mountain or a hill, somewhere with really high altitude. If you've ever been, hiking, or if you've been to like Ladakh or something, there people will say, right, that this is really high altitude, there's low oxygen pressure. If you're, it might make you sick if you're not used to it, right? You might need to take some cylinder or something with you. So why is that, right? And how is it that people live there and they're fine? So populations at high altitudes have been shown to have natural adaptations that actually reflect even in their genome. So they have genetic alterations that help them survive at high altitudes, right? How crazy is that? And people are just now, or like, with time and as technology improves and the way and the sort of efficiency and expenditure when it comes to mapping out human genomes, when it becomes easier and easier, we'll get more information. So these kind of adaptations in such populations, people at high altitudes, or in fact, I was even reading about it in deep sea divers, right? Deep sea divers also have these nitrogen imbalances. So how do they deal with it? And it's been seen that there's this population, I'm forgetting the name, unfortunately. If anyone knows, just you can just say it. But there's this population of people who literally, like, their whole ancestry, everyone performed like a lot of deep sea diving for fishing and all of that. And these people have actually like genetic changes that have been induced in them that allow them to be really good divers and not feel that crushing pressure and lack of oxygen to that degree, like what you and I would feel. So these kind of things are super interesting. Anyway, going into the details a little more. So this place, if you can recognize by any chance, these are the Andes Mountains. Okay, and they're a super long continental mountain range. As you guys would know, they're in South America and they extend along the western coast of South America. And it's super high above mean sea level. So you can see 4,000 meters above mean sea level. So just for comparison, like Bangalore, where I am, is about 1,000 meters above mean sea level. Um, Delhi is about 300 meters. Mumbai, Chennai, Kolkata, they're all at 0 meters above mean sea level. So this is really high. Okay, so if you live on at this kind of height, you need to have some kind of adaptation or an oxygen cylinder constantly strapped to your shoulder, right? Another example is Tibet. So Tibet is a region, as you know, right, in the on the Tibetan plateau, which is really high, and it has an elevation of like more than 4,000 meters again, right, in the Himalayas. And so people living in Tibet, they have to have some special way to survive, right? So it turns out that there is. So this is from a paper that was looking at these populations and looking at what kind of differences, um, what kind of like oxygen stress people over there are experiencing, right? So at 4,000 meters elevation, which is both of these places, every breath of air that you take in only has 60% of oxygen molecules in comparison to the same breath at mean sea level. So where you're sitting, you're breathing 100%, but if you go onto the Tibetan plateau or you climb the Andean mountain, then you have only zero point 0.6 times of that. So every breath you are accumulating that lack, right? So this is a constant feature of the ambient environment, and every single person is exposed to it. Now, if you have less oxygen in inspired air, you have less oxygen diffusing in the bloodstream, you have less oxygen going into your tissues, and you have less oxygen, therefore helping with energy production and metabolism in the mitochondria, right? So, and since humans don't actually store oxygen, we can't do that. It just reacts really rapidly. So there has to be some kind of supply. We really need it, right? And if you look at these graphs, oops, if you look at these graphs, you can see that this is inspired oxygen, but then this is the sea level. This is at mean sea level, and this is at a certain altitude. And then this is, if you look at, this is the altitude for inspired air in on a mountain. So at this height, and this is how low the oxygen tension is in at these different regions of either it's in the air or in the blood in comparison to the sea level. So you can see how to read these graphs and how intense it is. So there were these two adaptations that were these two populations that were studied, Tibetan and Andean natives, and people looked at what genes have, had changes and so on. These are a bit old, but the, it's still legit, right? So the two high altitude populations can be viewed as the current outcome of separate episodes. So they are here. They're saying that okay, both of them have adapted to high altitudes, but in slightly different ways. I mean, that's irrelevant right now. But anyhow, uh, that means that that they beat natural selection in some way or the other, and they improve their function and they preserve the genes that help them survive and 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 uh, and 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 beat high altitude hypoxia, right? And so some strategies that they use are like elevated ventilation, so you breathe more rapidly or deeply, so that increases your uptake. You can have lower hemoglobin concentration, so you can have in comparison to Andeans, you would think that someone would need to have more hemoglobin to overcompensate for this. So but that could actually cause viscosity and buffering problems in the blood. So actually, they have higher oxygen saturation of hemoglobin, which transports better, and some other things. And there's actually some genes like this one that were found to have certain uh, um, point mutations and changes in them that allow for these that are presumed to help these physiological traits and stuff. Yeah, and there's actually some increased capillary density and so on. So all of these actual changes and strategies were taken over in during natural selection in these populations when they were surviving in hypoxic conditions. Okay, so that was just an aside. I thought it was interesting, and I hope it was useful slash interesting for you guys as well. So I'll jump right into some questions on some basic concepts, and as we discuss the questions, we can recap anything that you guys may have forgotten or you have any confusions about, right? So as I was discussing earlier, which primary mechanism allows the blood to direct more, allows the body to direct more blood towards tissues with higher metabolic demand? So I was talking about how the body is clever and decides where to send the blood based on the state of the body. So, do you guys know what the answer is? For those of you who haven't attended before, you can just send the answer that you think it is in the chat in this meeting, and we can discuss. So, does anyone know which one it is? Okay, does anyone else want to try? Okay, so I have two answers so far, and while the correct answer is A, I also agree that D is partially correct. So let's talk about why that is the case. So the answer here is A, which is if you want to direct more blood flow towards a tissue, what do you have to do? Right? So suppose you want to direct more blood flow to the skeletal muscles because you're running. So you will have vasodilation in those arteries. Vasodilation is what? It just means that you're increasing the diameter of that blood vessel, so allowing more volume of blood to move through it. So vasodilation of arterioles via smooth muscle relaxation, right? So you guys learned about smooth muscles in the that are backing these blood vessels, right? So that smooth muscle relaxation is what allows vasodilation of the arterioles, and that allows more blood to flow into the tissue. But if you have vasodilation somewhere, then automatically that means if you're redirecting blood from somewhere, that means you have vasoconstriction somewhere else. Now you don't have vasoconstriction in venules per se, so maybe that's not entirely correct, but D option is sort of partially correct. Okay, so that I agree with. But yeah, the correct option here actually is A option, right? But you guys understand, right, that if the blood is being redirected, so you do have vasoconstriction in arterioles in the gut, for instance. All right, let's discuss the next question. So this August Crowe's concept of active and inactive capillaries primarily explains which of these? So we were talking about sphincters and capillaries and being able to decide, um, like the capillaries have some are active, some are inactive, right? And it's all part of a clever plan to make sure that the right amount of blood is going to the right organs and tissues. So with that in mind, anyone? No. Hmm. So the right answer here is D option, D for dog, right? Which is recruitment of capillary beds based on tissue demand. So it's not, I can see why it might be confusing with B, which is structural reinforcement of arteries, but the structural because of metabolic demand. And while there may be metabolic demand, there's no structural reinforcement that happens when it in arteries because you are changing the activity of capillaries and whether they're um, uh, transporting blood or not. So you have tissue demand that recruits certain capillaries to be active and certain that are not. So that's the concept here. I hope that's clear to everyone. Okay, so then let's look at the next one. So if you have an experiment where blood flow to the hand is occluded briefly with a cuff, occluded just means stopped, then the increase in blood flow after release is called which of these? Anyone? So the answer here, is reactive hyperemia, which is option A. So it's basically like an overshoot in blood flow that you'll get if the blood supply to a certain tissue was just blocked temporarily, right? And then if it was released, you have this overshoot. So you have occlusion, so the blood flow is reduced or stopped, which is called ischemia. And then you have a buildup of metabolites, you have vasodilation. Then, if you have vasodilation in that area because your body is trying to overcompensate, because suddenly there's less blood in that region, so you're vasodilating in the arterioles, and then that leads to the occlusion to be and then you release the occlusion, suddenly there is this excess amount of blood. So it increases above the baseline and then it eventually becomes normal as the metabolites that build up, like carbon dioxide or potassium and adenosine, lactate, right? You guys learned about that, right? That there are when um, when there's a lack of blood flow, there can actually be a buildup of these metabolic products like carbon dioxide, hydrogen ions, ADP, AMP, adenosine, and potassium ions, right? So in these cases, if you occlude the blood in your hand, say using a one of those sphygmomanometer cuffs, so the blood pressure measuring devices, they put like a cuff, right? So that is causing local vasodilation of your arterioles, and then you release it. Blood is gushing through now, and that is called reactive hyperemia. Okay, so I hope that is clear to everybody. All right, now next question. Uh, a decrease in oxygen saturation of blood typically produces what kind of effect on local blood flow? So this is the other way around. This is, anyone know? Think of it like a compensatory mechanism. Do you guys know what that is? It's just a way of trying to bring things back to equilibrium or homeostasis. So if you know, please send the message in the chat of which answer you think it is. Anyone? Today you guys are a lot more sleepy, huh? Anyway, so the answer here as a compensatory mechanism, right, is option C, which is increased blood flow. Why? Because if you have decrease in oxygen saturation of blood, what would happen? The body would overcompensate and push more blood there, thinking, okay, more blood means more hemoglobin, which is carrying more oxygen. So the body would then increase blood flow and perfusion of oxygen into that area. Okay. All right. Which of the following molecules is least likely to be released by oxygen-starved tissues to promote vasodilation? So I was just listing these. So if you guys remember, anyone? There are some metabolic products that are released by tissues that have some extra oxygen demand, and these then lead to vasodilatory effects that can increase blood flow and have efflux of all of these bad things that are accumulating and clean them up, right? So these local changes are, so the right answer here, I mean the wrong answer in fact, is nitrogen gas. You don't have nitrogen gas buildup, but you do have release of adenosine, and we'll talk about adenosine in some time. You have carbon dioxide, which makes sense, right? Oxygen starved naturally, but cells are going to respire anyway. So you have carbon dioxide building up and potassium ions, which are also building up, right? They're released from the tissues. And when these are detected, they actually cause vasodilation. Okay. All right. Let's move to the next question. Okay. So in the kidney, why is it that the afferent arteriole is usually wider than the efferent arteriole? Now, if you guys remember in the kidney structure, which you guys will, I guess, study in more detail in future lectures, there is an afferent arteriole that brings blood to the nephron unit, And then you have the glomerulus, which is a bunch of interconnected vessels where filtration takes place. So you have, and then you have an efferent arteriole that takes the now filtered blood out and then moves it to the proximal and distal convoluted tubules. If you guys remember that, I think we learned it in school, right? Afferent arteriole, glomerulus, efferent arteriole, PCT, loop of Henle, DCT, collecting tubule, right? And there's a lot of filtering and, sodium and chloride balance and water influx, efflux during these things. But why is, and you also must remember in your diagram that the afferent arteriole was typically, it has a higher a diameter than the efferent arteriole, right? But why? Why? Anyone? So, right. Okay, cool. Oh, so you guys were answering. My bad. Somehow I wasn't getting the notification for the messages. Right. Okay, okay. Thank God. Uh, cool. So does anyone else want to try answering? Yeah, exactly. Great job. So the answer is indeed B option, which is to increase pressure for ultrafiltration. Why? Because if you have, so ultrafiltration is a process that's taking place inside the glomerulus, right? And the glomerulus has these really thin capillaries where if you have high pressure um, passage of these of blood, then it'll force all these excess water and metabolites to leave, right? So if you have a lot of blood entering through an afferent arteriole, which has a very high diameter, you're suddenly shoving a lot of blood, a huge volume of blood, into very thin capillaries. So that means they're entering the glomerulus under a lot of pressure, right? So that is why it is important for ultrafiltration. Okay, and then it's leaving the efferent arteriole. It no longer needs to. It's no longer under so much pressure, so it can be a thinner or lower diameter blood vessel. Okay. All right. Let's tackle a few more questions. So you guys would have studied about renal autoregulation, right? So this primarily protects the kidney by which of these mechanisms? Yeah. So great job. I hope others can also try to answer if you're able to do that right now. I think many people also might be listening, but then they're also maybe eating dinner or something. So it might be difficult to send a message. Anyhow, as long as you're listening, that's important. So option A is correct, as said by you guys, which is maintaining relatively constant filtration pressure despite any systemic BP changes. So it was mentioned by, the professor in your coursework earlier on that while all of these different changes, with the help of all these hemodynamics mechanisms, they're changing the tissue pressure in different tissues, based on if you're running or if you're walking or if you're sitting or sleeping, you have different BP changes and adaptations. But your kidney relatively filters at a constant rate unless someone has some disease, right? So this is how renal autoregulation is the mechanism by which the kidney manages to retain and regulate filtration pressure at a constant level despite any other changes. Now, the other options here, right, which is, I mean, I'll paraphrase, which is changing sodium excretion, right, or preventing sodium release, right? These things, they are also to some degree part of the process. Like the kidney can selectively hold back on throwing out sodium and chloride, because where sodium and chloride goes, water goes, right? So if you want to retain water for whatever reason, you do change, the kidney does change how much salt it throws out. But that is not the primary mechanism by which it's protecting uh, uh, filtration pressure, right? It's not about filtration pressure. That's about water retention in the body, right? So anyway, that's just a perspective that that's another regulatory mechanism that the kidney does perform. It's just not correct in this context. Right. Okay, next question. So which receptor subtype mediates relaxation of vascular smooth muscles in response to adenosine? So remember, adenosine is a metabolic product that is released by tissues when they feel that there is low oxygen. And then adenosine goes and binds to a certain receptor, which is called a purinergic receptor. Why? Because adenosine is a purine, right? And it should not be roaming around outside of the body, outside of the cell, sorry. Yeah, so I'm seeing answers. I'm happy to see your answers. And has someone raised their hand? Yeah, do you have a question? You can unmute and ask me. No? Okay, maybe on accident. Right, so the correct answer here is C option, which is the A2 receptor. So adenosine, this metabolic product that's released in oxygen-starved tissues. Oh, you don't understand. Which one is it? Is it number 8 that you don't understand? I'm explaining that right now. So I think if it's not clear, then you can ask again. Yeah. Right. So adenosine is released by oxygen-starved tissues. Okay, and it is typically, it should not be outside of the cell. Adenosine is a purine. It's incorporated into DNA, right, or incorporated into ATP. So why is it roaming around outside? So it's like a cry for help, essentially. So when adenosine is released, it binds to purinergic receptors, right? And these purinergic receptors can be present in two places. They can be there. So these are all in places of muscles. Why? Because there needs to be some muscular response that allows more blood to reach the place that has oxygen starvation, right? So what would that mean? You would need to do vasodilation, or you would need to pump more blood with the help of the heart. So just in answer to that, body is so smart. We have purinergic receptors in SA node, right? Or sinoatrial node in the heart, and in smooth muscles. So including your vascular smooth muscles. What does that mean? In your blood vessels, you have smooth muscles that allow for this for this contraction to take place. So in the SA node, you have this A1 purinergic receptor, and in smooth muscles, you have the A2 purinergic receptor. Okay, and so in this case, the question asks, which receptor subtype mediates relaxation of the vascular smooth muscle in response to adenosine? So adenosine will bind to the A2 smooth muscle purinergic receptor, right? And that will lead to an increase in the level of camp inside the cell, which then leads to relaxation. Okay, so your question is explain the concept of adenocrine. What is adenocrine? Are you talking about endocrine? Um, I'm not sure, Shivani, about what you're asking actually. If you could just write out your question in a little more detail, then I'll try to answer it. Yeah. Adenosine only. Okay, so I explained it just now. Was that fine, or would you like me to explain some more? Adenosine, the major role. I see. Okay. So, all right. So normally, um, very little adenosine actually accumulates in the cell, right? So adenosine, where have you heard of it, right? Adenosine, for instance, in ATP, adenosine triphosphate, adenosine diphosphate, right? So adenosine typically, because you constantly have ATP being generated and consumed, you don't have just free adenosine hanging around much. Now what? So under conditions of homeostasis, where you don't have low oxygen in a certain tissue, you have a lot of ATP cycling and you don't have much free adenosine. But what happens if there is oxygen deprivation or there is a low level of oxygen in a certain tissue? So what happens is this oxidative phosphorylation, basically a process of generating energy with the help of oxygen, that slows down, and all the ATP that's there, that is consumed, but you're not able to make more ATP, right? Because your metabolism is not happening properly, it's slowing down. So you have ADP and AMP building up, right? And then you have some enzymes that actually convert this AMP. So ATP is adenosine triphosphate. You lose a phosphate, it becomes adenosine diphosphate. You lose one more, it becomes adenosine monophosphate. And now you have some enzymes that will convert your AMP to adenosine. So essentially now you have adenosine as a toxic product building in the cell. And then that diffuses out of the cell. There is a type of receptor, I think it's called a panaxin receptor. Uh, but I may need to check that. So that kind of receptor, it allows adenosine to move out of the cell. And this is not normal. This is only under oxygen starvation conditions, right? So that's the typical role of adenosine, and it's just part of making ATP. And yeah, and it shouldn't be outside, but once it is outside, it's a sign that there's low oxygen, and so you need to restore it in some way. Okay, so that is through these adenosine uh, receptors, adrenergic receptors. Sorry, not adrenergic, purinergic receptors. My bad. Okay, so I hope that's clear. Okay. Um, all right. So let's move to the next question. If you are still confused about anything, you can just explain. You can just put your question here, like you did. So that's great. Okay. Now, activation of A1 receptors. So this is again your purinergic receptor A1 type in the heart results in which of these events? Right. So I'm getting some answers. Hmm. The right answer here that I have in my notes is B option. Okay. So that is like negative chronotropy and reduced conduction velocity. So let's see why that is the answer, right? So if you have activation of A1 receptor, what does that mean? That means somewhere there is low oxygen and you have this adenosine binding to the A1 in the SA node. This actually leads to a reduction in the chronotropy. So that's negative chronotropy. So what does that mean? You have a reduced heart rate and you actually have a negative dromotropy, which means you have slower conduction. Hmm. So why is that the case? So that is one thing, right? That if you have purinergic receptors, based on where they are, you can actually have a different effect. Hmm. I'll get back to you guys. I think I am also a little confused about this one. Okay, we'll come back to this one. I'll just think about it. Meanwhile, you tell me what you guys think is negative dromotropy. Anyone? Yeah. So you guys have got it right, which is negative dromotropy refers to a reduced speed of electrical conduction in the heart, right? So that's something you see when adenosine binds to the purinergic receptors in the SA node. So that's just slowing of the heart essentially. So that's dromotropy. Now, which factor primarily regulates the normal rate of breathing under resting conditions? So this is based on like what kind of sensors we have in blood vessels and in the body. Ah, okay, okay. Right. So you guys are right. This is option A. So I was just reading because I myself got confused, which is about the previous question, right? Which is why is it that when adenosine binds to A1 receptors in the SA node of the heart, why is it that the heart rate decreases? So why is there negative chromotropy and negative chronotropy? Right? It's because you have to think about where this adenosine is coming from, right? Adenosine binding at SA node in the heart means that there is adenosine being released from heart muscle. That means heart muscle itself is oxygen starved. Now, if heart muscle is oxygen starved, what does that mean? That blood in the coronary arteries is not nearly enough to supply the heart, right? Maybe there is a block, maybe there is some, some other kind of problem, and the blood in the coronary arteries is not enough for the heart tissue itself, heart muscle itself. So there's not enough oxygen really reaching the heart tissue from the coronary arteries. So what's happening then is the heart cells themselves are releasing adenosine. This adenosine is then binding to the A1 receptors, which is a purinergic receptors in SA node. And that is then, now if to counteract this, the heart started beating faster, then even it would become worse, right? The heart would become even more oxygen starved. So this is like an autoregulatory protective mechanism that allows the heart to protect itself from oxygen starvation. So if the heart itself is getting low oxygen, the heart will try to beat slower. That's why when adenosine binds to A1 receptors in the SA node, that actually triggers a slowing of the heart so that it can compensate for the fact that for whatever reason there is low oxygen coming to it. Okay, so apologies for getting confused and confusing you guys, but that's how it works. Okay, so it's so cool, right, that you have an opposite effect on, cyclic AMP, and opposite effect on relaxation versus contraction, where in the case of smooth muscles, there is a relaxation that leads to increased blood flow, whereas in the SA node there is an opposite effect on cyclic AMP, which is your messenger, that leads to the heart slowing, right? So there's less blood going to the rest of the body, but blood being conserved for the heart. Cool. All right, I hope that's clear and I didn't confuse you too much. All right, so let's come to the next question before we go on to some topics. So why is it that all capillaries in the heart typically are active rather than selectively recruited? This is a pretty straightforward question. Yeah, I think this one is pretty clear, so I won't spend too much time on it. Yeah, great. So the correct answer here is A option, which is the cardiac muscle requires continuous oxygen supply. Your heart is, ideally it doesn't take a break, so it needs continuous oxygen supply. So whereas capillaries in your tissues may have sphincters that allow selective activation of capillaries, cardiac muscles and the cardiac coronary arteries and the capillaries associated with them don't have that joy. They have to continuously supply blood. Cool. All right. So now with these 12 questions, let's talk a little bit about this mechanism revolving around nitric oxide, right? So you guys learned about nitric oxide, right? And how nitric oxide synthase or NOS is uh, uh, um, part of responsible for transforming um, arginine to nitric oxide with a byproduct of citrulline, right? And this is in response to acetylcholine binding, leading to certain steps, eventually activating NOS, leading to nitric oxide. Now, nitric oxide just diffuses, right? And eventually, after some steps, it leads to muscle relaxation. Okay, so there are some in between steps of cycling, and then eventually that leads to muscle relaxation. Okay, right. So, okay, right. Oh, sorry. So now, just to go into a little more detail, right? So what triggers nitric oxide production, right? So it could be acetylcholine, like you can see here, right? And it also could be something called shear force. So what does shear force means? You guys might have heard about shearing or, or bulk shear and these kind of terms, right? Shear modulus, right? Shear stress, these kind of things. Essentially, it's like when a liquid is moving against some surface, and the movement of that liquid and the associated tension or stress inflicted on that surface, that is called shear force. Okay, so if you have blood moving along, and maybe it's too much, right? Or you have acetylcholine that comes and binds to a receptor um, on the endothelial cell. The endothelial cell is the cell that lines that forms the inner lining of the blood vessel, right? In that case, then what happens is you have this cascade where you have calcium, intracellular calcium levels increasing because they're released from the ER. This activates calmodulin, which will then activate the enzyme NOS, or NO nitric oxide synthase. This works on the raw material arginine, right? That's just an amino acid. And then you get nitric oxide, which is a diffusible product. So now what happens to nitric oxide, right? It activates guanylyl cyclase, right? And then you produce cyclic GMP with the help of GTP, right? And this then activates protein kinase G that has a feedback which allows the calcium level to come back down. So you don't have continuously high calcium. So it feeds back to the calcium, and it also leads to relaxation of blood vessels, meaning you relax the muscles in the blood vessels, leading to dilation of blood vessels. Okay. So this process was explained, and now we can tackle some questions relating to it. Yeah. So, all right, here's one of the questions. So, nitric oxide, responsible for vascular relaxation, right, is primarily synthesized in endothelial cells by which enzyme? This one is a dead giveaway. Yeah. So, let's not spend too much time on this one. I think you got it. It's option B, nitric oxide synthase, or NOS, right? Yeah, great job. So, now let's look at the next one. What is the immediate precursor molecule required for nitric oxide synthesis? So this also we just covered. Yeah, yeah, good. So the correct answer here is arginine, right? So arginine is the precursor molecule that NOS works on. Okay, all right. So I think that was covered, so I don't need to explain that again. Okay, so now after being produced in endothelial cells, nitric oxide reaches vascular smooth muscles by which process? Yeah, so you have to remember that nitric oxide is very freely diffusible, right? Basically like a gas. So the correct answer is it's option C. It just diffuses across the cell membrane. It doesn't need any active transport. It doesn't need any transporters, GPCRs, whatever, uniport, antiport, doesn't need anything. It's just diffusion, free diffusion across cell membrane. So that's why it's so potent, right? Because it can also travel pretty far. So yeah, that's how nitric oxide gets to the vascular smooth muscle. It just diffuses across. Right. Cool. All right. So let's look at one more question. So in vascular smooth muscle, nitric oxide directly activates which of these products? So again, I explained this earlier. Yeah, so I think everyone has got the answer correct, which is option B. So, which is soluble guanylyl cyclase. Okay, so that is what allows the level of CGMP to increase. That then feeds back to the intracellular calcium level, which decreases. Meanwhile, the smooth muscle relaxes. Okay, that allows vessel diameter to increase. It decreases the resistance to the blood, so increases the blood flow to that particular tissue. Okay, yeah. Also, interesting fact is that nitric oxide has like a half-life of only a few seconds. So it doesn't diffuse that far. I think earlier I said it diffuses far. I meant to say it diffuses near, which is that it will not go that far because it doesn't last that long. So it will just go to the region that's the most required. So you don't have systemic relaxation, because that would be such a problem if you have low oxygen felt in your leg, but suddenly like your arms are like getting extra blood, right? So that doesn't make sense. So that's what helps the specificity of localization of nitric oxide, is that it stays within a region because it has a very low half-life, short half-life. Cool. And drugs like nitroglycerin, they work by releasing NO. So that helps in patients who have angina, which is like pain in the chest because of a problem with blood circulation or enough blood reaching via coronary arteries to the heart muscle. Or if someone has some like heart failure or some, acute heart problem, you can give nitroglycerin that releases a whole bunch of nitric oxide that allows blood vessels to dilate, therefore allowing more blood to reach the heart. Yeah, cool. Okay, next question. So, activation of guanylyl cyclase increases levels of which of these, which then promotes relaxation? So, I mentioned this earlier on. So, this is just a revision. Hmm. So, actually, the the correct answer here is D option, but I can understand why it is confusing. It is CGMP, right? That is the currency here that's allowing for this feedback mechanism. Okay. So the correct answer is D option, even though CAMP is also another secondary messenger, but they are not interchangeable like this. They can both actually cause smooth muscle relaxation, but they're from different families. So they're different stimuli trigger CAMP and CGMP. So nitric oxide and stuff that triggers guanylyl cyclase, which then moves via the secondary messenger CGMP, whereas things like beta 2 agonists and all, they will trigger CAMP via adenylyl cyclase. Okay. So why is it that nitric oxide is specifically talking to guanylyl cyclase instead of adenylyl cyclase? One might ask, right? So that's because the nitric oxide is a gas and it is lipid soluble, right? It's uncharged, it diffuses, it doesn't bind a membrane receptor. Instead, it diffuses straight into the cell, and that is where your soluble guanylyl cyclase is as an intracellular enzyme. Okay, so it is specifically designed intracellularly to sense nitric oxide, and there is no kind of analogous nitric oxide sensing adenylyl cyclase that we find in human cells. So it is only possible via CGMP or guanylyl cyclase and then CGMP, right? So that is just that's just the physiological design of it, I guess, or the molecular design leading to this kind of physiology. So I hope that's clear now. The correct option here is D, which is CGMP. Okay, great. All right, let's talk about the next one. So the primary mechanism by which CGMP causes smooth muscle relaxation involves which of these steps? So this one is a little confusing and I can understand why. Okay, so one of you has got it right and the other has not. So I'll explain this so that you'll never confuse this again. Okay, so what is the goal of uh, this nitric oxide and signaling via CGMP, right? The final goal, physiological goal, is to cause vasodilation. Vasodilation needs what? It needs smooth muscle relaxation. If we want to relax muscles, so think back to skeletal muscles, cardiac muscles. What was the case of intracellular calcium when we were contracting the muscle? What was happening? Cytosolic calcium was increasing like anything, right? You had calcium coming in from outside, from the extracellular matrix. You had calcium coming in, being released, effluxing out of the sarcoplasmic reticulum, and the intracellular cytosolic calcium level was increasing like mad, right? In that condition, you have contraction. And then as the calcium decays, you relax. So in this case, if you want to cause muscle relaxation, you want to lower intracellular calcium availability. What happens at the molecular level if you lower calcium availability? Right? It does not displace troponin C and displace tropomyosin, therefore. So you don't have actin myosin binding, so you don't have contractility, right? So if you want to relax the muscle, you actually lower intracellular calcium availability. And that is the goal. And that is the goal is muscle relaxation, and the method to reach that is reducing the amount of calcium. That will lead to less contraction, right? So that's the correct answer here. Okay, it's option C. So good. I hope that is clear. Um, we can move to, I think I have like two, three more questions in this set. So, right, which physiological stimulus is well known to enhance endothelial nitric oxide release? So I think this one I explained right and at the get-go. So this one shouldn't be a problem. Yeah, perfect. So that's absolutely correct. It's shear stress, which is basically just the way the blood is going to rub against the endothelial cell and the force with which it's going to do so, that can actually trigger. So that's like a mechanical feedback, right? Think about it, right? It's not just a chemical sort of ligand receptor cross-linking reaction. It's actual mechanosensing. And there are receptors known as mechanoreceptors or mechanosensitive receptors that transduce this kind of mechanical information and have conformational changes in response to mechanical changes that they detect in the environment that then allow them to trigger uh, signaling in cells, right? So you have two types of inputs here. You have a mechanical input and you have a chemical input with like the binding of acetylcholine or something, right? Cool. All right. So now this one about a certain drug called sildenafil. This promotes vasodilation mainly by which process? Yep, that's the right answer. It's option C, which is inhibiting phosphodiesterase 5 to prevent CGMP breakdown. Right? So what do we want with the help of this drug is to increase blood flow, right? So cyclic CGMP, there's a specific phosphodiesterase that degrades cyclic GMP, and CGMP we need, right, for this vasodilation process. So um, if we have excess of this enzyme that's degrading CGMP, then we're not going to have a continued vasodilation. So if we degrade it, then CGMP persists for longer, which allows vasodilation to persist for longer, which allows blood to flow for longer. And if you know what sildenafil is, it's Viagra, so you know where the blood is going. Okay, so let's go to the next question. Endothelial dysfunction that reduces nitric oxide bioavailability is most strongly associated with which of these problems? Hmm. Great. So yeah, the answer is D. The confusion with B I can understand. So vasodilation is going to be excess vasodilation would be would not happen here, no, because if you are reducing nitric oxide bioavailability, then actually you would have the opposite of excess vasodilation. You would have excess vasoconstriction, or at least you would not have enough vasodilation when required. Why? Because nitric oxide is required to stimulate uh, relaxation of the vascular smooth muscles, leading to vasodilation, leading to more blood availability, right? Now, if you have endothelial cell dysfunction for whatever reason, then what's happening? Enough nitric oxide is not being produced or it's not being made available to the blood vessel muscles anymore. So then you don't have the correct amount of vasodilation as per requirement. So B option, it's actually the opposite of B option. So the correct answer here is D, which is hypertension and atherosclerosis. So why is you guys must have heard of atherosclerosis, right? It's a buildup of these sort of plaques because of cholesterol and so on and so forth. And then there are these foamy cells that really make a mess over there and have a lot of inflammatory secretion that leads to narrowing of the blood vessels, which leads to pain in the heart. If this is happening in a coronary artery, it can lead to heart blockages, angina, right? Oh, my voice is breaking. Oh dear God. All right. Hold on. Can you hear me? I'm just trying to see if I have some problem with my internet. Just let me know.
Yes, ma'am.
It's fine now. Okay. Okay, fine. All right. Good. Let me know if it gets weird again. Right. So atherosclerosis and all can cause this reduction of nitric oxide bioavailability. In the case where your endothelial cells make basically the endothelium of your blood vessel is clogged or it has some plaque buildup or something. Okay. All right. So now we'll come to a really, really important regulatory system. Just a second. Okay. So, right. A super important regulatory system called the renin-angiotensin-aldosterone system. This is like a multi-organ regulatory system. Renin is, coming from your kidneys. Then you have angiotensin converting enzyme coming from your lungs. Aldosterone again uh, um, coming from your adrenal gland, and then all of them coordinating together to maintain water balance in the body and pressure in the body, blood pressure in the body, and modulating the release and absorption of ions, water, and so on. Okay, so I thought it would be nice, instead of you continuing to hear my voice so much, is that I'll play a nice video I found, which very nicely explains the concept. Very short, I think it's just 2 minutes or something. So this will summarize it really nicely, and then we can look at some questions and discuss if there's anything you're confused about. Okay. So if you can't hear this, just let me know. I'll start playing it.
The renin-angiotensin-aldosterone system, RAS, is a hormonal system that controls blood pressure. While baroreflex is a short-term response to sudden changes in blood pressure, RAS is responsible for long-term regulation. In the kidneys, within the walls of afferent arterioles, there are specialized cells producing prorenin called juxtaglomerular cells. Upon activation by a drop in blood pressure, prorenin is cleaved to form renin, which is released into the blood. Renin converts a plasma protein called angiotensinogen, produced by the liver, into angiotensin 1, a peptide of 10 amino acids. Angiotensin 1 is further converted into angiotensin 2, an 8 amino acid peptide, by the angiotensin converting enzyme, ACE, predominantly present in the lungs and kidneys. Angiotensin 2 is a hormone. It binds to angiotensin 2 receptors in tissues to exert various effects. It stimulates vasoconstriction in systemic arterioles. It promotes sodium reabsorption in proximal convoluted tubules of the kidneys. It induces the release of aldosterone from the adrenal cortex. Aldosterone promotes sodium and water retention in the kidneys. In the central nervous system, angiotensin 2 has several effects. It acts on the hypothalamus to stimulate thirst and encourage water intake. It induces the posterior pituitary to release antidiuretic hormone, which promotes water retention by the kidneys. It reduces the sensitivity of baroreceptor response to increased blood pressure, so that this response would not counteract the effect of RAS. All these actions lead to an increase in blood volume and blood pressure. Angiotensin 2 is short-lived with a half-life of 1 to 2 minutes. It is degraded into angiotensin 3 and 4, which have lesser effects. Overactive or inappropriately activated RAS is a cause for hypertension. RAS is a frequent target of antihypertensive drugs. ACE inhibitors and angiotensin receptor blockers are common treatment for hypertension.
Okay, great. So I hope that was clear. We'll of course discuss it further as we go through questions. Uh, okay, so noise. Is someone saying something? Does anyone have a question? Right. Okay, so then let's start with some questions relating to the renin-angiotensin-aldosterone system, and we will discuss as we go. Okay. So renin is secreted by which kidney cells, specialized cells in the kidney? So you can answer in the chat for those who have joined a little later. You can text it into the chat box in the meeting. Yeah. 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 So the right answer here, this is just like a sort of theory question. There's not really any big why to why like the answer. So the answer is C option, which is the juxtaglomerular cells. What does that mean? It's just cells near the glomerular apparatus. Okay. So those are the guys that secrete renin in the kidney. Okay. All right. Let's move to the next question. Which condition most strongly would stimulate renin release from the kidney? Do others want to try? What do you guys think? Could it be option A? Could it be option B? Could it be option C? It's definitely not option D, huh? So, the correct answer here, most of you got it, and those who didn't, I hope now I'll make it clear. It's option B. Why is this? So, what is the, again, you have to, when it comes to these kind of things, no, you have to remember what is the goal, okay, ultimate goal of this pathway or this system. The goal is to respond to reduction in blood pressure. So, it's to regulate blood pressure and increase it as and when required. Okay, that is the whole goal of renin-angiotensin and aldosterone system. So in option A, if you have elevated arterial blood pressure, that's the opposite of what would stimulate renin release, right? Because the goal here, and renin release is the start of this RAS pathway, right? The goal here is to increase blood pressure. So elevated arterial blood pressure, that's not what's going to do it. Reduced renal perfusion pressure, though, that is the correct answer here, option B. Okay. So what does that mean? Perfusion pressure? That just means it's the reduced pressure of blood flowing through the kidney, right? So if this is detected in the kidney, then you have a stimulation that leads to renin release. And then that activates the rest of the pathway. That leads to retention of sodium, retention of water, in the body that leads to increase in blood pressure so that it can come back to homeostasis in some way or the other. Okay, so the correct answer here is B option, reduced renal perfusion pressure. I hope that's clear now. Okay, all right, let's go to the next question. Renin converts angiotensinogen into which of these compounds? This one follows from the video you watched and your coursework. So angiotensinogen comes from the liver, and there's this angiotensin 1, right? Option C, as you guys have said. Great. Option C, angiotensin 1 is there in the plasma, and this renin works on it and converts it. On angiotensinogen, it converts it to angiotensin 1. Okay. So that is clear enough. We'll move on to the next steps and then discuss further. Okay. Angiotensinogen is produced where? So I unfortunately gave this answer away, but it would be good if you could answer it anyway. Yeah, fine. So the answer here is liver, right? Option B. So great. Now, this enzyme, very important enzyme, you heard of it a lot in the context of COVID also, right? Angiotensin converting enzyme ACE is found in the highest concentration on endothelial surfaces of which of these regions in the body? ACE enzyme, okay. Angiotensin converting enzyme. Yeah, yeah. So the correct answer is pulmonary circulation because it is released from the lungs, right? So that's what it's pertaining to. So ACE you will find mostly in the lungs or the pulmonary circulation. Okay, all right. Now, what is the most potent direct effect of angiotensin 2 on blood vessels? After this, I'll just recap once about how we got to angiotensin 2 and then we'll continue on. Yeah, yeah, I think this one is clear enough. You have vasoconstriction leading to increased peripheral resistance. Basically, you're trying to increase the blood pressure, so you're constricting, right? So that is one of the most potent. So angiotensin 2 is like a super strong kind of a signal here, right? So till now, what have we seen? We saw that the blood pressure drops, okay, and that is a signal to the body. It could be because of dehydration, right? You have not taken enough water, or you have salt imbalance. It could be because of hemorrhage if someone is injured and they're losing blood, so their blood pressure is falling. They have low sodium, as I mentioned, somewhere you need to drink some ORS or you need to take an IV drip, or some other cause leading to reduced renal perfusion, as we mentioned. So, renal perfusion pressure has decreased. This is detected by the kidney, and then the juxtaglomerular cells release the enzyme renin. Renin works on angiotensinogen, which is from the liver, and converts it to angiotensin 1. And angiotensin 1 is converted to angiotensin 2, this guy, with the help of this enzyme from the lungs, which is angiotensin converting enzyme or ACE. Now you have angiotensin 2. This is like this is some kind of like an emergency messenger, okay? And it manages to raise the blood pressure quite fast using different mechanisms, one of which is vasoconstriction, right? So it is quite powerfully causes vasoconstriction, increases vascular resistance systemically. So that means in the whole body, not locally, systemically, right? Because all of these reactions are happening in the blood, so it's going everywhere and it's binding to everything, right? And then it does some more things, which I will cover as we go, right? So it stimulates aldosterone release also. So aldosterone primarily acts on which part of the nephron? So if you think about what aldosterone is telling the kidney to do, that would make it clear as to which part of the nephron it is acting upon. Again, always keep the goal in mind. Keep what is this system trying to achieve. Right. So the right answer here actually is C option, which is the distal tubule and collecting duct. So why is that, right? So for this, you need to remember what does this kidney, like the nephron, what does it look like, right? So if you remember in the nephron, sorry, you have different regions after the efferent arteriole, right? So you have your proximal convoluted tubule, loop of Henle, which is this loop that goes downwards, and then you have a distal convoluted tubule ending in a collecting duct. Okay. And the blood that circulates around it from the glomerulus. So that is from the Bowman's capsule, and then the blood that goes around the glomerulus is what through which the absorption or efflux will take place, or reabsorption, as they say, will take place. So sodium reabsorption, which is what we need to do, right? Aldosterone is trying to do what? Again, we're trying to increase blood pressure. So for that, what do we need to do? We need to increase sodium retention or reabsorption into the body. So how does that happen? The kidney is capable of regulating this reabsorption, and in the region of in the region of the distal nephron, there is this tiny amount that actually allows to balance the sodium, because most of your reabsorption is happening in the PCT, and that is actually not touched. Actually, where it is, why it's not working on PCT is because in the DCT, there's such a small amount of reabsorption that takes place. If you directly work on that, that can actually balance sodium and balance blood pressure. The real reason as to why it's not acting earlier on in the nephron in the PCT and instead it's working in the DCT and onwards is that you'd actually end up reabsorbing too much sodium. So even a tiny change like this of amount of aldosterone in the blood could actually lead to way too much sodium reabsorption, leading to way too much blood volume or retention of fluids. And so that is not precise and that is dangerous. So the distal convoluted tubule, because it is on the distal part of the nephron, it's only responsible for a tiny part of the sodium reabsorption, but that is enough because again the body is never regulated by brute large, changes. It's always small changes that result in big effects, right? So in this case, if the DCT is reabsorbing a tiny amount, like 4-5% worth of the sodium, that's enough to retain enough water to then regulate blood pressure. And this is an analogy that I remember learning in physiology in college, which was wherever sodium goes, water goes. Okay, so if you're reabsorbing sodium, you're also retaining water. You're reabsorbing sodium, you're increasing blood pressure. Okay, so that's why aldosterone, I took a bit of an aside, but aldosterone primarily acts on the distal tubule or the distal parts of the nephron. Okay, so I hope that is clear now. That's also something I had recently sort of looked into because it's a bit not that intuitive, right? Okay. What is the main physiological effect of aldosterone that contributes to high blood pressure? So this I already answered. I'm not going to waste time on it. Yeah. Yeah. So you guys have got it. It's an increased sodium and water reabsorption. So you are retaining all of these via the distal parts of the nephron and the kidney. And aldosterone comes from the adrenal gland. Okay. So that's just, I don't think I have questions on that, but that's just to remind you. Okay, and then it works directly on the kidney and it increases sodium and water reabsorption, leading to increased blood pressure. Okay, now suppose one were to use an ACE inhibitor. By what mechanism would it lower the blood pressure? So you have to remember, what does ACE work on? And if we took it away with an inhibitor, what would happen? Yeah. Yeah, so this is pretty clear. Yes, great. Everyone has got it right. I'm happy about that. So it's option B, which is blocking the conversion of angiotensin 1 to angiotensin 2. Okay, so I explained this earlier, right? That ACE is the substrate for ACE is angiotensin 1, and then once you have angiotensin 2, that is the real sort of chief currency of the RAS pathway. It makes everything go really fast, it changes blood pressure really fast. So ACE inhibitors do this. Okay, I think we have just 2, 3 more questions. So, which medication, I wonder if you guys know, I don't know if it was mentioned in the coursework, I think it was, is a well-known ACE inhibitor. This is just a bit of trivia, you don't need to worry about it. Okay, so the correct answer here is D option, enalapril. Okay, so enalapril is a very commonly used ACE inhibitor and it, yeah, it's used often in the clinic. I mean, I don't know too much about it. But it inhibits ACE and leads to decreased angiotensin 2, and it actually also, I think, affects like bradykinin levels. And as if you remember from your coursework, if you have increased bradykinin levels, that can lead to vasodilation as well, because it can stimulate the endothelial cell, which would then talk to the muscle cell and so on. So you if you increase bradykinin level by inhibiting ACE, you can actually cause more vasodilation. So I think these two effects it has. So I hope that is clear. Uh, yeah. So in case you want to know what the other ones are, right? So losartan, I think it's a very well known drug. It is an angiotensin 2 receptor blocker. Okay, so it blocks it and it prevents angiotensin 2 from acting. So this would also stop your blood pressure from increasing, but it doesn't inhibit ACE, right? So it directly acts and makes it competitively inhibits out angiotensin 2. So you have the same effect, which is that you have vasodilation, and you have decreased, whatever levels of aldosterone and lower blood pressure, but you don't have the bradykinin level increasing. So I guess some doctors would prefer it over enalapril, right, for certain reasons. And then propanolol, that is it's a beta blocker, right? So you guys heard about beta blockers, right? So this is very important when it comes to regulating heart and sympathetic system. If people have if you block the beta 1 and beta 2 receptors, you can essentially decrease heart rate and contractility. You can decrease renin release. And yeah, so it's used a lot for patients who have hypertension, arrhythmias, I think heart pain, like angina and stuff. And it's quite versatile. It's used a lot in these kind of problems. Yeah, so it's different from losartan and enalapril, uh, but it's used in a similar context. Cool. All right. So that's some extra trivia for you. Okay, two more questions. So second last question is ACE also normally breaks down which vasodilator peptide? Oh no, I gave the answer. Hmm. So the correct answer here, I'll just rush through it now because we're anyway reaching the end of the session. So the correct answer here is bradykinin. Okay, so I was mentioning how ACE inhibitors have two effects, right? One is that it inhibits ACE, which then directly affects the level of angiotensin 2. It is a decreased level of that. And it also, ACE normally also breaks down bradykinin. But if you inhibit ACE, then bradykinin level goes up. Now, what does bradykinin do? It binds to receptors on endothelial cells, which then lead to nitric oxide production and relaxation of blood vessels. Now, if you inhibit ACE, what will happen? With an ACE inhibitor, for instance, bradykinin levels will continue to increase. So then nitric oxide, so it's like you're tricking the body, right? You're just causing bradykinin levels to increase. Bradykinin is binding more and more to endothelial cells. Therefore, more nitric oxide is being produced. More nitric oxide is flowing through the tissue into the smooth muscles of the blood vessel, and that is leading to more vasodilation. So that's what ACE does. It breaks down this peptide to regulate vasodilation, but you can mess with that with an ACE inhibitor. Okay, last question for today. Blocking the RAS pathway, for instance, using these medications we talked about, would result in which of these overall effect? Yeah. So now I think that is clear then that the motivation of the RAS pathway is to increase blood pressure. So if you block it, then it's option B, you reduce peripheral resistance and lower the blood pressure. Okay, so that's what a lot of these inhibitors like ACE inhibitors and losartan and stuff, that's what they do. Okay, all right. So I think that is all I have. Yes. So as usual, if anyone has any questions, please do send. You can always contact me via the common portal, I think. I'm also uploading these to YouTube. I think you know, so you can like leave comments there as well. If you have questions, I'll try to answer them. Um, yeah. So I hope this was useful to all of you, and yeah, and that you join next time, and we'll continue with the next week's coursework. Okay. Someone is joining now. Okay. All right. Yeah, you're welcome. It was a nice class. All right. Great. Okay. All right. See you guys next time. If anyone has any questions, don't hesitate to reach out to me. I will try to answer them as the best I can. All right. See you next time then. I'll leave.